Hello and welcome. My name is Mitch Sanders. I'm the former chair of the Communications Committee, and I'm here to host a new series with Jordan Yarn to feature awardees of the Wound Healing Society annual meeting. And so our special guest today, we have Kanhaiya Singh. He's assistant professor at the Indiana Center for Regenerative Medicine and Engineering at Indiana University. Kanhaiya, welcome.
Thank you.
Jordan, before we get started, because you've been an active member of the Wound Healing Society for a number of years, you've been on the Communications Committee for a number of years as well. It'd be great for people to kind of get to know you and the great work you're doing at ASU as well. So could you provide just a little bit of background on yourself first as my host of this new series, and also your research direction and what you're focused on?
Absolutely. My name is Jordan Yarn. I'm an assistant research professor at Arizona State University in the School for Engineering of Matter, Transport, and Energy. And we're studying both fundamental and translational aspects of wound healing, from understanding novel mediators of wound healing to developing drug-eluting wound dressings, trying to turn a chronic wound acute and solve this whole big problem, or try to. So it's a pleasure to be.
That is really cool. We need to sign a non-disclosure agreement. We have big pharma clients that we're doing drug elution studies, and we're doing some really cool things, but I can't tell you all about what we're doing until we sign an NDA, but we have to chat after this call. Kanhaiya, congratulations on winning the Excellent Translational Science Award for the Wound Healing Society. Can you tell us more about your epigenetic work you did in DFUs? And all the studies that got you here to win this award?
Thank you. First, I will thank Wound Healing Foundation and Wound Healing Society for this award. And the title of this work that was presented in Wound Healing Society meeting was Endothelial PLCG2, the missing link that make VEGF therapy robust. So as we know, and as research says, in diabetes, there is a condition called vasculopathy, in which there is low blood flow in vessels. And this is because there are some secondary metabolites like advanced glycation end products or continuous hyperglycemia that lowers the responsiveness of endothelial cells towards hypoxia or towards VEGF signals. So in this work, we are looking at how this VEGF therapy can be augmented in diabetic wounds or diabetic ischemic limbs. So for that, this was quite a new study for us, because we used single-cell RNA sequencing for the first time in our lab on human skin samples. So I will just update you about single cell. That means you can measure the transcriptome profile of a single cell at a time. And as you know, the human skin is made up of 15 different compartments. You can go and query each and every compartment at individual level. For this example, we queried endothelial cells from diabetic and non-diabetic skin samples. And we sequenced around 36,000 cells, individual cells, out of which like 2/3 were from diabetic and 1/3 were from non-diabetic. And then we proceeded with only endothelial cells. And we found there is another cool technique called cell chat analysis, in which you can measure how one cell type is communicating with other. And we found that in diabetic skin samples, there was no much change of VEGF ligands and receptors communicating with each other. So that raised another question. So what is different then? So then we looked into the differential expression profile, and we found a molecule called phospholipase gamma C, which is a VEGF signaling pathway molecule. And we validated that. We found that yes, through immunohistochemistry also, that level was down. So then we thought that if we can somewhat augment this PLCG2 gamma or PLCG2 gene in diabetic skin we can rescue some of the functions, mainly angiogenic functions in diabetic ischemic limbs. So we used acute model of hind limb ischemia in this case. And to deliver these PLCG2 molecule, we used our own tissue nanotransfection technology. Okay, so this is a technique in which you can deliver your cargo inside the cells without disrupting cells a lot. That means you can have the functionality as well as you can overexpress the gene of interest. And we found that yes, after we overexpressed PLCG2, the hind limb ischemia was getting rescued. But it was not in the case of diabetic limbs, because in diabetic there are certain other things that may compromise that. So then we try to give VEGF therapy in addition to this PLCG2 or overexpression. And then we found that yes, this VEGF augmentation with PLCG2 was indeed giving more beneficial effects of this rescue from hind limb ischemia. And then we also did some like mechanistic study where we knocked out PLCG2 and then we gave VEGF. We found that this VEGF effect on rescuing from hind limb ischemia was gone. That means certainly VEGF therapy has something to do with PLCG2. So this was the work that we presented. Of course, there are a lot more mechanistic details that are presented in our published paper, which is just published in Diabetes Journal in May issue. So one may find it interesting to look over what is going on.
Wonderful. And what are you doing next? What's your next big?
Yeah. So next thing is it was just a cause and effect. Now we are going into the mechanism, how it is happening. And I'm pleased to announce that I just got a DoD Discovery Award to do that. So like, as you mentioned in the start, epigenetics. So diabetes is an epigenetic disease. That means microenvironment in which we are in that affects the genetic environment too. So we are looking into how PLCG2 is getting repressed in diabetes. And one of the reasons can be hypermethylation of that PLCG2 promoter. So now we are trying to demethylate in vivo using our same tissue nanotransfection technique. So this is my next goal, how to specifically demethylate and then see how it can rescue diabetic wounds, maybe.
As a nerd who's more focused on commercial, did you file a patent on therapy of PLC2 and VEGF as a combination therapy?
No, not yet, but I'm in planning.
Before you present the data, you have to file a provisional.
Yes.
Otherwise, other people can take your work and call it theirs almost. So
yeah.
So anytime you have a really good idea like this, which is a very good idea, write a half-page invention disclosure and submit it to your tech transfer office and say, hey, we should write a patent. Hey, the paper's going to come out. Hey, we're presenting the abstract at WHS. That way you can protect the IP, and then you'll get a trickle of royalties from the university that'll be yours. But this is a really cool idea, and it should be investigated as a drug. Now, the cool thing is Big Pharma's coming back into wound care, which is a huge surprise to me. And this could be a real interesting opportunity. But we've learned so much over the last 30 years why many drug trials failed. A lot of it was because the design was poor. We didn't have a run-in phase. We didn't remove the wounds that were healing on their own but also we've got much better, smarter at designing these trials so we can get better data and better outcomes. But this is a low-hanging fruit that should be picked up by somebody. But next time, when you have such a great idea, please file a provisional patent and protect it so that you'll be able to capture a little bit of the wealth and excitement of working with a commercial partner. And that's all I do every day.
thank you for this idea. I will do from next time. So yeah.
You're going to have another great idea tomorrow. So write a half-page invention disclosure and send it to your tech transfer office. What do you think, Jordan? Anything else?
No, I second that idea of protecting your IP. It's sort of the first thing that we do over here is we're sending out disclosures all the time. But I'm going back to your science. I got very excited by what you said about the GC2 alone not being sufficient, and the VEGF alone not being sufficient, but together a synergistic activity. And I'm curious if this is the current topic of your investigation, or if you have actually elucidated what is that synergism that VEGF is doing that allows the phospholipase to be active.
Yeah, so this is my next question that I'm working on, that how signaling molecules, VEGF and downstream signaling molecule all together is passing via PLCG2. So I'm doing some lipidomics research, how this the signal is getting passed to the actual effector molecules.
Mm-hmm.
It's got to be some sort of phosphorylation cascade, right?
Yes.
So do you know the canonical target for this phospholipase?
Yeah, so it works via DAG, arachidonic acid, PGE2 mechanism. So prostaglandin E2, we all know that it is there in wounds, but how it is getting down in diabetic wounds, we need to more focus on that. So yeah.
So it sounds like it's targeting the inflammatory phase, the early inflammatory phase of healing. What would happen, or maybe this is a question that you have not yet investigated, so if this is, then patent it, I guess. What would happen if you waited until a later stage of healing to hit it with VEGF and the phospholipase when it's potentially out of the inflammatory phase and, let's say, arrested in the proliferative phase?
Yes, that's a good idea. So we should investigate on that. So yeah, so this was only prevention kind of model, but we should also look on the rescue model.
So very exciting.
Yeah.
Did you guys see Proma Besu's paper on the early phases of a chronic wound and using nanostring to examine the biochemical pathways that are turned on and off? It really got me very excited. I'm actually going to use nanostring both for wound care and I've got a program on traumatic brain injury as well. And sometimes when you don't know what question to ask, you let the cell or you let the cells tell you the answer, and then you can sort it through. And I think that that's such an elegant approach. And what's amazing to me, I remember how much time I used to spend on qPCR processing 384 samples or whatever it was. And now you send the RNA to them, and the next day they have the data. It just blows me away how we've advanced and how we can start to really tease out the entire biochemical mechanisms and how all these different pathways work together. It's really, really very interesting. So Kanhaiya and Jordan, I'm going to keep following your work. You're doing great stuff, and congratulations on the award. And thank you for being our first podcast recipient of the award. So this was really fun. We are going to ask a silly question, and then we're going to finish. If you were a superhero, what superhero would you be?
That's a good question. So I would be maybe Iron Man, who do not get diabetes, maybe because its external shell will protect him. So maybe. Yeah, but before I finish, I just want to thank my mentor and my center director, Dr. Chandan Sen, to help me in enabling to work like this with a huge enthusiastic group that I have. And yeah, so I will thank him for enabling these kind of research. Of course, we work, but we need some guidance too. So, and then the first authors of that paper, Dr. Rustagi, Dr. Abouhashem, Dr. Verma, and There are two Verma is like one is Sumit Verma and the other one is Priyanka Verma. So I will thank to them too.